Good morning, everybody. Wasn't that an amazing session? So I don't know if anybody else was out dancing last night. I'm looking around, I think they are, but I just, just in memory of a really awesome evening, I just thought this might be important to A, send the message, and B, bring down the lights just a little bit. So anyway, no, it's my pleasure to be here. My name is JP Clancy. I'm a pediatric pulmonologist and help oversee a lot of the clinical research programs in partnership with Patty Burks, who is our Senior Director for Clinical Trials Affairs and really makes the engine of the TDN go. So I am going to be kicking us off, talking a little bit about some of the, we're going to talk, I guess I'm going to be talking about the what, and Patty's going to be talking about the how. And yeah, hopefully that'll make sense in just a moment. And so, see if the clicker clicks. No, but we'll try this one. That works. So I'm not going to worry about the clicker. It makes me think of Last of Us anyway, and we don't want to go there. Anyway, three things. If you have to walk out of here, three things to remember. One, highly effective modulators are changing CF before our eyes. And I think it's sometimes hard to sort of appreciate how rapidly CF is changing when you're sort of in the middle of it. But it is remarkable and unprecedented, and we're right in the middle of it. And I'll give you some interesting new findings around modulators for those who are eligible and have participated in clinical trials. But it's astounding. Second, we are going to continue to need therapies to treat the symptoms of CF. And there's a number of reasons, particularly including people who are not modulator candidates because of their genetics or they don't tolerate modulators, and also because a lot of people who are on modulators have damage to their lungs and they have other symptomatology that goes on. So we're going to need symptomatic therapies going forward. And then, probably most important, reminding us that the rare and nonsense mediated CF group, they're going to be the first in line for the genetic-based therapies. And again, you had a nice sort of introduction of that today, and we'll talk a bit more about that. But Patty's going to start talking about some of the realities of trying to do those studies. It won't be business as usual. So I'm going to give a very quick reminder of what CF is, because I know people have different levels of experience and connection to CF. CF is caused by a problem with the gene that codes for a protein, and a protein is something that goes and does a function, does something in the cell. And that protein is called CFTR. You can see its full name there. I won't say it again because it's long and complicated. When it doesn't work, you get the symptoms and, unfortunately, the disease, cystic fibrosis. We know that about 1 in 3,000 people in the U.S., roughly, those numbers keep changing, are born with CF. We know that over 40,000 people in the U.S., well over 100,000 worldwide, have CF, and it infects a lot of organ systems. And I have them listed here. And the point of it saying it's not just one thing, it's a number of things. And the way we approach and treat those different organs can be somewhat complex. And it also just reminds us that the disease itself, as Chad told us yesterday, just manifests in very different ways for different people. If you looked in the CF airway, and we looked at sort of right on the surface of those cells, we would find that CFTR, its job is to move salt and water. And when it's not working, salt and water don't move the way they should. And what does that mean? It makes mucus thick and sticky and hard to clear. And that mucus problem actually is pretty similar throughout the organs that are affected by CF. Certainly some differences, but by and large, thick, sticky, hard to clear mucus is a problem. And in the airways, that leads to a place where bacteria and fungus can come in, set up shop, not get cleared, cause chronic infection, and the symptoms, and over time, damage to the airways. And again, that infection part maybe not so important in other organs, but clearly is, for the lungs, is sort of the primary cause of morbidity symptoms and, unfortunately, shortened lifespan. So we're going to talk a little bit about modulators. What are they? Well, these are they're little molecules. They're basically drugs. They call them small molecules because they're of a certain size. And they will go to that and bind to that CFTR protein and help it work more correctly or fold or get in the right shape, basically. And what we have here is an example on the left side is a CFTR that's not working. It's up where it's supposed to be to do its job, but it's stuck, it's closed. And that's what happens actually with the G551D CFTR. And on the right side, see those little kind of, I guess they're kind of like little orange blobby things? Those are supposed to be modulators, and they're binding to different parts of CFTR, helping it get in the right shape so it can open and allow the salt and water to move the way it's supposed to. And so that's kind of the key point. They bind to the protein, that thing that is actually performing the function, and help it work more normally. So, fast forward 10 years from when the modulators really first hit clinical trials. And this is just looking at results from Trikafta in people who have one copy of F508. Trikafta was developed in the laboratory over a number of years to really help that most common cause of CF, the F508 mutation, work more normally. And what we're looking at here is looking at lung function on the top. And the blue are people who were treated with Trikafta during the study. You see this whoop, super fast jump in lung function, and it stays there for at least the 6 months of the study. And the orange is people who are on placebo, basically an inactive medicine. And as you see, very steady, not much change. And if you look down below, same timeline, same people, but we're looking at the sweat chloride test. And what we see is if you're not on Trikafta, sweat chloride stays high, stays right around 100. And for people who were on the arm receiving Trikafta, a rapid drop in sweat test actually below the diagnostic level that we have for CF. So huge changes across, and that's just working on one of the CFTR, just that one gene, the protein that came from the one gene, not two. So very effective. This is a little more recent data that came from a big study that's ongoing. It's called PROMISE, and it's basically looking at outcomes for people who started Trikafta as part of their clinical care, and it's looking at a variety of different outcome measures that were not looked at during the clinical trial. And this is more recent data in children who started Trikafta and rolled into this PROMISE study. You can see their mean age was about 9 years, about 125 people. And what we're looking at here are sort of 3 tests that are not routinely done in the clinic. The first is something called a lung clearance index. That's basically like a super sensitive pulmonary function test. It works really pretty well in kids because it's really good at picking up changes, even if the lung function test, FEV1, looks very normal. And what we're seeing here on the left side are each one of those lines is a person in the study, and the orange, kind of orangey red thing, that's the average of them all. And what we could see is there's this whole scatter on the left side. And the higher you are, the more abnormal you are. So what we're seeing is even in this young population, the dotted line being the normal value, there's a fair number of people who started out with abnormal lung clearance index. And as you see, after 6 months of treatment, you can see the vast majority of them have actually dropped into the normal range. Not everybody, but certainly a great deal of them. In the middle is looking at the changes in sweat chloride again in this group. Again, by and large, moving from the around 80-85 range down to the dotted line. And the dotted line is actually normal. Not just CF diagnosis, that's like below that is considered normal. And so a lot of people getting below normal, the average getting right about to it. And on the right side, this is looking at symptoms by a test or a questionnaire that we commonly use in clinical trials. And what we see on the left side is a wide scatter and an awful lot of people going up, and that basically less symptoms, higher number. And it's not perfect. We realize there are symptoms going on, but I kind of circled up at the top. There's an awful lot of people up there who actually were not having any more symptoms. So again, exciting data that sort of reinforces what we've learned from the actual clinical trials. Another thing that was looked at PROMISE is looking at bacteria, and specifically we know that a number of different types of bacteria can cause symptoms in CF. And this is looking at the density or the amount of bacteria that could be detected in the mucus. And as you move from left to right, you go before you started Trikafta and then different times at sort of 1 month, 3 months, 6 months, 12 months. And each one of those lines is a person. And we're looking at Staph aureus, Pseudomonas, something called a Chromobacter, and another one called Burkholderia. And the point is, the red line is the average. You see that all of them went down. And in fact, if you look on average, it's about a 100-fold drop. We're looking at a log plot, basically, but the amount of change is amazing. It's really even more than we see with antibiotics, believe it or not. And so this work is still ongoing. How long is this going to last? But the point is, you see this dramatic effect on infection in people with CF. A lot of people actually clearing infection. And this was really looking at an older group, 12 and above, and the average was in the 20s. We spoke about the Simplify study that was highlighted by Steve in the last session, and this was asking a very simple but kind of profound question in CF. Can people with CF who are doing well and stable on Trikafta, can they start reducing some of their treatments? And the way they did this, it was a really ingenious type of study, but it basically allowed people to basically enroll up to 4 times if they wanted to, and they could look at how their lung function performed, how it looked, how it held over 6 weeks of either staying on pulmozyme, staying on hypertonic saline, dropping pulmozyme, dropping hypertonic saline, and all those combinations in between. So people could spend time either on or off and serve as their own controls. And so a lot of people enrolled, almost 600 people, and a lot of people went back into queue, about 850 people. And an important point here is that the average lung function for this group of people was really good. It was 97% basically. So we don't know if this applies to everybody. But what we found, if you look on the right side, this is looking at the change in lung function. The blue was the group who either stayed on or stopped hypertonic saline, and the red are either the people who stayed on or stopped pulmozyme. And I'm not going to worry about getting into the circles versus the triangles because it doesn't matter. They all did about the same, and that's what's pretty amazing. Does this mean stop everything? Of course not. But it does mean that this is an open question, and it's it's time for conversation with care providers, and B, at least what you can take away is maybe that weekend trip I can sort of not bring everything with me like perhaps I had in the past. And again, we're going to be learning more. This is a short study, but obviously really exciting, and follow-ups will come. There are more modulators in development. Steve spoke about this, and I won't go into details of all of these programs, just to say, though, why is this important? Well, we know that not everybody has tolerated all these different modulators, the current modulators that are available. We think that some of these other modulators may have the potential to be more effective even than what we're seeing with Trikafta right now. We'll know with the clinical trials, but there is that possibility. And I also realize too, for people who have rare mutations that do make some protein, but they don't respond to something like Trikafta, maybe they could respond to some of these other modulators because they are different, and they're sort of different structures. What we're looking at on the right side is basically a cartoon of the CFTR. And each one of those little call-outs is an area or a part of the CFTR. And actually, the different molecules have been made to match these different parts. And the point is, you might find that the right combination for one person may not be the right combination for another person. And that's the world we're going towards by having more opportunities. So Steve highlighted that thinking about the future of CF research, we know there's a large group of people who are modulator eligible and are having great benefits. But there's also a significant number of people who are not modulator eligible, don't tolerate, have side effects, all these things. And for that reason, we need to continue to develop therapies that treat the symptoms that occur with cystic fibrosis. And also another point, we've talked about this, the CF community is growing. Whoever thought that was going to happen? It's sad to say my week-old slides are now out of date, apparently. Median survival, 56. Thanks for the heads up, appreciate that. Other exciting things, transplants have dropped precipitously. Babies, and more babies, lots of babies happening. But I think the point I wanted to make here is that most adults with CF will have some lung damage, and that adult population is going to get bigger. And what we're looking at here is just an estimate of what the population could look like over time. And honestly, we did this work back in 2016. It needs an update, but it's actually coming true. And so each bar is adults with CF at different time points out to 2031. And the color is a number of those who have either very normal lung function in green, have some lung damage in yellow, or maybe more advanced lung damage in blue. And the point is, the people that are living longer often actually have lung damage, and because of that, they're going to need ongoing symptomatic therapies. So this is a pipeline that people have probably seen. A lot of different therapies are under development across the spectrum of CF. Starting on the left are drugs that are therapies that are really targeted at bringing back or giving people CFTR in different ways. And I highlight the one in orange, infection, where it continues to be a problem and a high priority for people with CF. I can't go into great detail for all of these for obvious reasons, because Patty will, pull the hook. But the point is, I'll give you some updates for a couple of things that I think are of particular interest potentially, and happy to answer more questions. Anti-infectives, Steve mentioned phage. Again, this is the silent battle that's going on in the world and has been going on way before us and will probably go on way after us between bacteria and these little viruses that attack those bacteria. We don't really notice them, but point is, this isn't a new idea. This has been around over a century. What's changed now is we've really become much more capable of sort of identifying, selecting, growing basically, and then delivering a given phage. And again, a couple of companies who are doing work with some early results that are looking exciting. They'll be presenting at North American CF Conference. But point is, there's a real potential here. And if you think of an antibiotic as sort of a blunt instrument, sort of whacks down everything, a phage is a very surgical instrument. It just knocks down that guy and doesn't bother everything else. So you can see how that might be a good thing, because it's not going to hopefully have effects on your GI tract and all the other, things that can come with using lots of antibiotics. Second one I mentioned is nitric oxide, or NO. This is a natural molecule in our body, does a lot of things, but at high levels, it actually is very good at killing bacteria. And it can kill a lot of different types. It's not just this or that. It actually can cover a lot of bacteria. And an exciting study that we're helping support is looking at high-dose inhaled nitric oxide at home to try to fight non-tuberculous mycobacteria, or NTM, which is notoriously difficult to treat. So some real exciting work coming. Another concept that is in line with that is gallium. Gallium it's basically it's an element, or it's an ion basically, that is able to, it's like a Trojan horse. It goes into bacteria and substitutes for iron. Iron's really important for a lot of different processes in a bacteria. The gallium gets up there, messes up those processes, and actually kills the bacteria in lots of different ways. And so it has the potential to be sort of a whole new different way to treat infection. And we're helping support a study that's looking at inhaled gallium for Pseudomonas infection. Antibacterials, kind of more traditional pills and so forth to treat Staph aureus as an example. We're helping support the development of a drug from Acerigen, which is able to kill staph, including MRSA. And then finally, I mentioned an antifungal. We know that fungal infections are definitely a problem in CF, and particularly in people after they've had a lung transplant. And this study is looking at an inhaled antifungal to help prevent fungal infection after a transplant. Again, no results to report, but exciting results to come very soon. Finally, I'm going to just give you, I don't have to go deep here because we just talked about this, but Path to Cure, as you are probably aware, is really for everyone with CF, but currently is laser focused on people who are not candidates or not able to take modulators, and really is a push to bring people in and to make this move faster. There are, as we've sort of alluded to, several strategies to try to give everybody a working CFTR. Path to a Cure it does include modulators, we've talked about that. We've also included RNA and also talk about DNA, and each one of these has its own nuances and differences, but all of these are different strategies that we're undertaking with trying to have many shots on goal. There are at least 50 programs that are either funded, 35 specifically, and a number of others which are under evaluation. And they're all sort of different parts of a bigger ecosystem. Some are focused on restoration, some are gene delivery, some are editing, some are the tools for us to know that they're working, and a variety of other sort of novel approaches to sort of bring companies together. One who works on part A, one who works on part B. Together they make something better than either alone. And this slide, again, I find extremely exciting and terrifying, okay? Patty and I, a lot of our life is trying to strategize to develop and bring these exciting therapies into clinical trials to prove those which are effective and beneficial. And again, similar slide to what we saw before, but point is, there's just a ton of things either in clinical trials or just on the cusp of clinical trials. And now, the big question, how are we going to do this in a really limited population of people? And now we're talking, numbers 1/10 of what we normally do for clinical trials. So with that, I pass it on to my partner in crime, Patty Burks.
Thank you, JP. I think a lot of you already know JP, but I'll take a second and introduce myself. I'm Patty Burks, and I'm Senior Director of Clinical Trial Affairs at the Cystic Fibrosis Foundation, where I work closely with JP. His office is next door to mine. I try to keep track of him. I do my best. We do have fun. I came to Bethesda from Washington University School of Medicine in St. Louis, where for 9 years I worked as a cystic fibrosis clinical research coordinator and just learned so much there. What a tremendous staff they have. I don't know if we have any people here from St. Louis or from Missouri, but a great center there. Great center there. Before that, I was a pulmonary clinic nurse and a staff nurse at Children's Hospital. But the first time I ever heard about cystic fibrosis was when a pulmonologist at Little Rock Children's Hospital sat across from me and explained to me why my 8-week-old child couldn't gain weight and at such a young age already had a cough. I'm a mom. My son's life was full and rich and happy and wild and huge and big, but it was not long. And he passed away when he was 11. That was long before we had any of the drugs that you saw in the pipeline today. And the world has changed dramatically. But I just wanted to start off by saying it's amazing what a parent can do when they're dealt a pretty rough hand. And there are parents here in this room today who are quite amazing, and you are changing the world. So I just wanted to stop a second and say thank you. Let's partner together until it's done. Thank you. So back to the talk. Thank you, JP. That's where my notes start. Well, how are we preparing for this road ahead? We will build on success. We will listen well and do what's needed, and we will develop collaborations. This year, we celebrate 25 years of the Therapeutics Development Network, founded in 1998. The TDN has worked tirelessly to deliver speedy therapeutics and treatments to people with cystic fibrosis, resulting in the most significant therapeutic advances in the history of the disease. The network, the largest CF clinical trials network in the world, has grown in its number of participating sites and its reach across the country. This is one of my fun facts. In 1998, 11% of the CF population received care at a CF care center that was also a TDN site, providing this 11% access to participation in clinical trials there at their local center. Now, 59% of the CF population receives care at a center that is also a TDN site. But with the trial finder available on cff.org, our logistical and financial support of study participant referrals, and the development of the regional cooperatives, which we'll hear more about in just a minute, we're doing all we can to make clinical trial participation available to all who are interested, no matter where they live or where they receive their care. Over the past 25 years, the TDN has done what research networks do. They enrolled patients in both interventional and observational trials. They completed studies, and they made and accepted study referrals so patients could participate in studies not conducted there at their care center. They did all the things. You may wonder, how do you enroll over 34,000 participants in trials for a disease that most of this time, the population was only 30,000? The CF population in the US, which was the CF population in the US when much of this work was done. We enrolled more patients than we have. The CF community is unique in its generosity. Not everyone with CF is eligible to participate in clinical trials. Some are too young. They might not have the right mutation or the right bacteria or don't meet other eligibility criteria. And some, well, they're just too sick. But those who can enroll for those who can't. And then you know what they do? They do it again, and they do it again. And you know what happens? Outdated slides is what happens, JP. This is what happens. I'll just say, as a mom from the 80s, who'd have thought? Who'd have thought? Onward. And so there have been success, tremendous success, but there's more to be done. CFTR modulator drugs were surprisingly effective, but a substantial portion of the people with CF have mutations that would not respond or they were intolerant. We looked ahead and wondered what was next. I like that slide, it's kind of fun. And I think JP and others saw this situation coming a while back. I have to say, when I saw this, JP, I was happy, it's fun, and terrified. I was really happy to hear you use that word too. Okay. And so I believe JP and others saw this coming a long ways back, and so initiated in 2021 the Genetic Therapies Working Group led by Manu Jain and Chris Goss. One of the subgroups was responsible for developing the research education resources that our TDN sites would need in order to move into the genetic-based therapy space. It was my privilege to lead this effort along with my colleague Sharon McNamara, a research coordinator from Seattle Children's. We developed a plan for how we would move forward, a plan that started with a lot of listening. We listened to research coordinators and investigators, to those living with CF, and to external groups with experience in genetic therapies, and to experts, including scientists, researchers, and ethicists. Knowing that the population eligible for these early genetic-based therapy trials would be those with rare mutations who are not currently on modulators and who are more ethnically and racially diverse, more than the general CF population, we conducted a survey looking at recruitment practices of underrepresented minorities. This was sent to CF research coordinators, and the responses you are looking at are from those who have successfully recruited underrepresented minorities into clinical trials as they answered the question, what leads to success? Well, you'll notice here that transportation and printed materials in appropriate languages helped. They're very important. And again and again and again, we heard in our listening the importance of that trusted team member introducing the idea of research to the family. We conducted another survey to assess the educational needs of the research team members and then followed up with another survey a year later to see if we'd moved the needle. As we assessed the educational needs, we heard that the research teams wanted webinars, video recordings, printouts, and infographics. And we responded, and we conducted a webinar series in January of 2021. In fact, Dr. Kuzar, who's sitting here today, was one of the investigators who presented in that webinar series. We focused on the history and recent successes within genetic therapies, some of the unique recruitment considerations in genetic therapies, and then really spent some time talking about consent. And again, the unique considerations around consent when it comes to this kind of research. So I lost my place. In response to this, we conducted a webinar series and then developed a training roadmap on our TDN SharePoint site that is available to all research teams. This includes a select group of core trainings, an assessment tool, a certificate of completion, and in addition to this, we have provided a library of supplemental trainings as well. This is data from our follow-up survey where we asked about the respondents' personal knowledge on a topic and then about their comfort level in discussing this with patients and families. And you can see we moved the needle. There's still work to be done. We are not finished, but we are pretty proud that in a fairly short period of time, we were able to make some significant progress. We built on our past success. We listened and developed what research teams wanted and needed to be prepared for what was ahead. And then we built collaborations like never before. We're going to have to do this together. There's 288 care teams, 91 research sites, 40,000 patients and families. We have to do all we can to facilitate trust between these groups. And with that in mind, we built the regional cooperatives, a place where trust can be built. You will remember that 59% of the CF population receives care at a care center that is also a TDN site, but 49% do not. Many of these upcoming trials are small, with only a handful of our participating sites and enrolling only 16, 18, 20 participants. The regional cooperatives are part of our approach in doing all we can to make trial participation available to everyone interested. And so the goals of the regional cooperatives includes cultivating trust, building relationships, collaborating across CF care centers, research centers, removing barriers to communication, and to share best practices. And it is our hope that as we do this, it is our plan that as we do this, we will improve access to clinical trials and increase referral opportunities. Every care center, affiliate, and research site has a regional cooperative that they are part of. I have to just stop a second. This was not a small task. This is massive. And Kelsey Pearson, my colleague at the Therapeutics Development Network, has really done a lot of work here. I don't know if anyone here knows her, but she's absolutely brilliant, and this is a lot of her work. So we're well equipped and prepared for this road that we're on. We have a history of success. We've listened well, done all we could to prepare, all we can to prepare, and will continue to do all we can. We are working collaboratively like never before. And with our most important work ahead, we will not rest until it's done. Thank you.
There we go. Thank you, Patty. That was wonderful. And let me reiterate that it's been no small task. Seems so straightforward to say, we'll just have the care centers and the TDN centers sort of, it'll be easy. And yeah, there's yes and no. It depends on where you are and what the dynamics are on the ground. So it's a long way of saying we're early days. I mean, we started this work really just at the turn of the year, and I think we're gathering momentum. But really hope that this is a means that we can really improve the access and just improve the reach and the interest and ultimately participation in clinical trials. I have a question that came in. Is the CFF researching modulator effects on the developing brain, pediatric populations? Do you want to tackle that? I didn't think so. Okay. Sorry. It's a great question. I think a lot of families and care providers and researchers have wondered what is the relationship between some of the mental health effects that have been described for people on modulators and its cause. Is it because of the modulators? Is it a combination of the modulators and some of the things that Chad talked about yesterday and some of the others about sort of all of a sudden a whole set of new expectations put upon someone who has been given a 20, 30, 40 more years of life that they didn't see coming? Hard to tease apart, as you can probably guess. There are research programs that are, how we fund research is, we do it in different ways, but one of the ways is people will submit grants that undergo review by scientific experts that we brought in to do those reviews, and then fund those which are strong science and obviously are priorities for the CF community. And so this type of research is being funded in laboratories. I couldn't give you the details today just because it's not exactly our program. That's kind of more on the basic science side. But I also know that we're working with some of our other research networks. We help to support research networks in Europe, in the UK, in Canada, and also in Australia. And the point of that is that there's also been a great deal of interest in capturing mental health data in people who initiate modulators. And so we're working with those groups who are kicking off their own studies like PROMISE in younger populations so we can really capture that data. And the last thing I was going to say is that we do have a mental health research working group. It's a little different than what Bruce described. That was the Mental Health Advisory Council, perhaps a bit more care-focused. The mental health research working group was more research-focused. What are the questions that need to be answered by clinical research? And so we've literally just had gone through the application process, and they'll be having sort of their kickoff meeting this summer. So lots more to come, definitely an area of great interest and importance, obviously, for all of us in the community. So you don't have to do it on the, do your questions here. You're welcome to raise your hands if there's any that we could answer for you. Come on. Yes. I can come down. Oh, I want to do my Oprah thing. Come on. I like Oprah. Here we go. You get a card. Yeah, I knew that was coming.
You get a car. I just had a comment and then a question. Comment is, thanks for staying engaged with the fight. I think parents who lost kids in the early days of this and remain involved are heroes. So thank you. And then the question is, it seems like with all the new modulators in development, is it that, and for those of us who have kids on Trikafta and those kids are doing well, is it that some of the new modulators might be objectively more effective, or is it just providing optionality for those patients with additional treatments?
So I think everybody heard a great question. Are these modulators better, or are they just different? And I think it's a little bit of both, to be honest with you. I think we appreciate that people who are on, again, some people don't have a full benefit, and again, others have side effects, can have liver function test abnormalities, et cetera, et cetera. So having medicines that are truly metabolized different may be very beneficial for people. But also some of them look actually more promising if you look at sort of side-by-side comparisons in laboratory. And so, again, some of the stuff that's been shared, looking at some of the combinations from groups that I shared with you, Sionna, AbbVie, and also Vertex, with additional modulators under development, actually outperform Trikafta in the laboratory. Big question, does that translate into clinically feeling better? Can we measure it? Does it mean something? I think that's the big unknown. I personally think that until I believe sweat chloride's a pretty good readout for at least that the drug it's active. Granted, there's always a reason that maybe this organ doesn't see the full benefit compared with that organ, for maybe doesn't get into the levels it needs to be, or maybe there's underlying damage, or take your pick. But the point is, I think until everybody's sweat chloride is indistinguishable from people without CF, there's progress to be made. And so anyway, is too much CFTR a problem? Yeah, it could be. Honestly, cholera, that's a problem where CFTR gets turned on and it doesn't turn off, and we know that's very problematic. But thankfully, nothing that's been in the laboratory has been we haven't seen that kind of concern. So anyway, but it is both. We want more options, and we want better than the current options. So yes.
Thank you. Is there any research going on currently, or have you seen any data speaking to patients on modulators who have long-term cultured positive for NTMs and now are not? And is that something to look forward to as a continued benefit of Trikafta, or is that something that may come and go, or is it individual to the patient? Sorry, loaded.
All about the NTM, right? First of all, so NTM, for those who don't know, stands for non-tuberculous mycobacteria. It's a type of bacteria that is found in roughly 10-ish% of people with CF, maybe a little more. Not everybody that you detect it actually has what we call disease, where the bacteria is actually creating signs, symptoms, and damage. However, for those who do have NTM, and there's different flavors of NTM, one of them that's particularly hard to treat is called abscessus. The treatment regimens can involve 4 or 5 antibiotics, prolonged side effects, and even with that, there's still a number of, good number, I mean, over 20, 30% who may not clear the infection, and even higher in some conditions. So there has been reports that Trikafta, one thing it definitely does is reduces how much sputum is produced, and so there's a lot less detection of NTM per se, but is that just a sampling problem? And we're honestly in that figuring out phase in regards to that. I don't think I've seen data that would clearly say that NTM is being cleared by Trikafta, but I also acknowledge it may be early days. Jen Taylor-Kauser works here at National Jewish, which is the biggest NTM care facility in the country. So please correct me if I'm wrong, but I'd say it feels like we're sort of in the figuring out phase. Does this really represent clearance, or does it represent just not detecting the same way? So, and I think the other big question, which is unknown, is what will it look like over time? If ivacaftor or Kalydeco, which is another very highly effective modulator for a subset of people with CF. If that's any predictor, we would anticipate benefits will persist. However, those who've been on Kalydeco, if we look at data 5 or 10 years out, there is some loss of lung function over time. So I think we have to be very vigilant as we look forward with Trikafta, because I don't think we're convinced yet that stability is going to stay. I think we're hopeful, but we don't know that yet, to be honest with you. Oh, yes.
Let's see. When mRNA trials begin, will enrolled participants taking modulators be able to enroll in them?
Yes. Well, it's a great question. I think it depends. Different programs have gone about it in different ways. I don't think there's an absolute FDA guidance on this question yet. A lot of the design features of these early phase clinical trials are sort of made in collaboration or in conversation with the FDA. mRNA, in theory, is not going to be terribly long-lasting, and it doesn't really carry risks that I would say of perhaps some other genetic-based therapies. In other words, the mRNA is transient, it's going to get cleared, you should get back to baseline over time. And so for that reason, maybe the risks for someone who was on a modulator may be considered tolerable, I guess is the way to look at it. That may not apply for some other genetic-based therapies where you actually might end up having a true change in the genes of the person receiving the therapy. And that's a really interesting space, and I just, maybe Patty, I can turn it over to you to think a little bit about it. One thing we're learning about some of these genetic-based therapy approaches, depending on the type, the FDA requirements for follow-up can be as long as 15 years. And so, anyway, what does that mean to you, Patty, as a research coordinator? Because this is not simple.
It's very challenging when you start thinking about the numbers of patients that would be eligible for these trials. And it sounds like in the early phase trials, it'll be more likely that they won't be on modulators, at least these initial ones. So that's a very small group. And then I already talked to you briefly about eligibility criteria in a trial. There's all kinds of age and, all kinds of things that have to be met. So we really are getting to small groups, almost like a needle in a haystack. And just when I think about that, JP, I just think collaboration. This won't happen in someone's, garage. It takes all of us working together, patients, care centers, and research centers.
Yeah, and I guess the only other thing I would say, we really want to set up the ecosystem to let people participate in repeated studies, that has been a big part of the success to date, is being able to have people who are willing and able and find the time and have the capabilities to participate in clinical trials to help participate. We don't want those long-term follow-ups to get in the way of being able to, if you're in study A, it's done, unfortunately didn't work or didn't achieve its goals. Now study B is available. We want to make those A people to still be able to do B, and that's new territory, to be honest with you. And getting the FDA comfortable with that, it's going to take a little bit of time. Yes, sorry, here we go.
You actually answered part of my question, but for somebody who's on Trikafta that's already in advanced lung disease, if their lung function drops, will these new medications, it's like antibiotic, can you become resistant? And will the new medications help?
Yeah, so kind of the question around would could some of the medicines that help to restore CFTR function, be it a modulator or a genetic-based therapy, could somebody sort of develop resistance to that? I think that's probably wouldn't be resistant in the sense that, well, let me think about it a second. I would say there's a risk that you could have, let's say you receive a genetic-based therapy and that genetic-based therapy triggers an immune response. And then your body may actually, if you get redosed, it may actually sort of inactivate that second dose. And so we're paying a lot of attention to that. They're called, an example is something called neutralizing antibodies. They're basically things floating around in our blood, and their job is to detect things that are not us, that are foreign. And it turns out that genetic-based therapies are foreign, and we're trying to design them so that they don't send off those signals, those alarm bells for the cells. But it is a risk, it is. On the other side, though, if somebody has advanced disease, or perhaps even not getting full benefit from a modulator for a reason that sometimes is the difference between person A and person B. If I have allergy symptoms, I get a lot more out of Allegra than I get out of Zyrtec. I don't know why, but that's not the same for everybody. But the point is, there may be a place where these inhaled CFTR therapies could add on top of what we see with the modulator. And so for those who aren't getting the full benefit, perhaps we can also push that benefit more by adding on a genetic-based therapy. Oop, over here. They're going to keep me running. There we go. Sorry.
I've heard from multiple people who are post-transplant that they can't take the modulators even though there are mutations that make them eligible. So I'm wondering why that is, and if there's any research or work being done into looking into modulator options for those people post-transplant.
Yeah, it's a great question, and it's true. It's definitely an observation. When people have had a transplant, those lungs no longer have CF per se. I mean, they have the CFTR of the donor person. So, but there are still lots of other manifestations of CF that are not improved by a modulator, GI manifestations, sinus manifestations, variety of others. And I think that unfortunately, it's not been a slam dunk to start a modulator and those other symptoms get a whole lot better. It's about a third to a half of people who try modulators post-transplant choose not to continue them because they're not seeing benefit. Or maybe they're even feeling side effects. I don't think we fully know why that is. I mean, one possibility is that most people who've had a transplant have more advanced disease in other organs too, and perhaps those things don't reverse as clearly. And I will say also that modulators, while clearly beneficial for a lot of symptoms, haven't been beneficial for everything. It's been hard to show that they help the GI symptoms. And whether that's because, as an example, maybe your gut microbiome gets set at an early age, and it's hard to change that when you're 30, 40 years old. I don't know. We're still trying to figure that out. But the point is, not everything in CF gets fixed by a modulator. And we're finding that with the post-transplant population.
Yeah, this is a quick question related to the FDA. It's sort of a follow-on for what was the earlier question. In terms of just the numbers, like in the phase 3 trials for novel treatments, assuming people are not also on, let's say, a modulator and just getting one of the newer RNA, is I know we have a great relationship with the FDA, but then there's statistics that are significant when you're doing clinical trials. I understand that. And so the question is, do we have an overall concern if we're going to looking at a very small subset of people, in other words, smaller numbers within that small number of population, in terms of getting valid data that is clinically significant?
Sure. Yeah, big question. How do you do clinical trials in this ultra-rare population? Patty, do you want to tackle any? I'm happy to follow up, but I know you think about this a lot.
Well, I think of the struggles at the sites. You have to build relationships with this population and trusting relationships. And the first time you come and speak to someone, it really shouldn't be about participating in a genetic-based therapy trial. So there's opportunity for what we've been doing for 25 years through the Therapeutics Development Network is to develop a culture of research among this group. But I'll let JP take it from there.
Well, the other part is but first of all, is having the culture beyond the borders of our TDN sites, and that's really a big part of it. That's increasing the quote that N. But I guess the other point too is the nature of the clinical trials will be fundamentally different for genetic-based therapies. Genetic-based therapies for other rare diseases can be approved with as 50 patients. The numbers are a lot smaller. And some of those may not have a placebo group during the study, believe it or not. And that seems kind of crazy, but when some of these other disorders, I'll give you an example, spinal muscular atrophy, it's a horrible, incredibly difficult disease where children, from birth, very soon after birth, really start losing strength, and honestly, death is usually in the first 2 years of life. That study, I think, got approval with 25 people, able to rely on what they say historic controls. Clearly, you have to have a pretty good, clear benefit to be able to follow that path. But we're really trying to position ourselves to be able to do this by looking about sort of very different trial designs where perhaps there's a shared placebo group, or even we develop data from a group of people who are not candidate for modulators now that we can then share across different clinical trial or different clinical development programs. And so then you're talking about one placebo group, and they also get to participate in the trials as a historic control. So I can't say that's all tucked in at this point, but it's definitely the direction that we're going, and it's not unique to us. It's actually how things often progress for ultra-rare diseases like this. So thanks, great question. Oh, I'll get there in a second.
So I don't know if I'm going to ask this correctly, but there's 3 families within my chapter that were in the Trikafta studies. And I think you know about this, or I sent you an email maybe, I don't know. And they did better on the compound that didn't make it. And so what kind of guidance can I give them? Who owns the compound? What are they doing with it? Will it be in future modulators? I mean,
Yeah, I think there, and as people may remember, during the development of Trikafta, there were sort of two side by side, two combinations that had one difference between them, and some people seem to do better on one, and some people seem to do a little better on the other. And I can't necessarily speak for Vertex, but I think they made the decision to go with what was called 445 as opposed to, I think, 659, which anyway. Based on just the side effect profile, in the majority of people, there seemed to be a little better on the 445 side, but that's not, universal. So it's still their compound. I think what I would say though is the drug that is under development right now, I actually don't have the full name yet. The newest triple, the once daily, does have 2 molecules that are different than the original, and so I think there's the potential for people who may have seen a step off from moving from the 659 to currently Trikafta may actually see some benefits there. So it's a fair question. I couldn't tell you 100% though, and I'm not aware of them trying to advance that other 659, but I do think there is interest in continuing to make better combinations and better iterations of the current drugs. There's one back here somewhere. Here we go.
So we're familiar with people who understand they're carriers of CF and not exactly exhibit the disease. Is genetic therapies a conversation for carriers, and is that something that is talked about at all?
Yeah, interesting question. So would carriers be candidates? Probably not out of the gate, certainly because thankfully, the vast majority of people who are carriers don't have CF. I guess there's always exceptions to any rule, but generally one has to have both copies not working, being it's a recessive disorder. So if you have one working, that's enough. But is it possible that there would be in a world where people would correct that second one? I guess it's there. It just has to be really good. It would have to be really, really precise and perfect to be able to fix that. And you'd obviously want to, that would be a ways down the road. But thankfully, the vast majority of people who are carriers certainly don't have CF. But they are, if you look, the people who are carriers do have a higher, as a group, a higher incidence of sinusitis, higher incidence of actually male infertility, higher incidence of subtle symptoms of CF. So I guess I would say I'm careful not to say no, but it would have to be in a kind of unique scenarios, tell you the truth, probably. Teresa? Sorry. They did not make it easy for the Oprah thing, but that's okay. I needed my steps. Here we go. I'm getting my steps.
Just a question for you, since we're having trouble making sure we have enough trial participants, what about thinking internationally, and what are the challenges and opportunities, and how does the FDA think about that?
Yeah. Well, do you want to talk a little bit about how we work with our global partners? And I can talk a little bit about the regs.
We are partnering with our global partners, and there's a number of protocols where the sponsors approves an international review, so that when we're reviewing the protocol and the safety of the compound, we can do that together with UK and Canada and Europe. So we are working together. That would be my first statement. Then beyond that, you have to be really careful. So one thing that people have been interested in is, what if we bring people in to participate in a trial here in the US, but they're ex-US? That there's ethical considerations about bringing a patient in to participate in a trial when in their country they have no hope of accessing that compound. So that's only one of the considerations. There's a whole host of others, but those are my thoughts. JP, what do you think?
Well, I was just going to say, yeah, we're definitely partnering internationally. This is definitely a global effort. And we hosted a number of representatives of research networks across the globe in the CFF offices 2 weeks ago, having our global therapeutic development meeting. And I think it was extremely, I was extremely pleased, can't come up with a better word, that everybody was on the same page that we have to do this together. And I think the main reason for that is, if you get one message from this group and one message from that group, you sort of have developed this ecosystem where people are sort of, bouncing around and not moving forward together. So there's definitely a desire for unity in how we do this. And also, interestingly enough, what I didn't really appreciate is the variability in different countries in the modulator ineligible population. And just where I'm going with that, our colleagues in Europe, just highlighted country by country. Less than 50% of people in Italy and Spain are modulator eligible because of rare or different mutations, which just kind of blew my mind. I don't think I'd ever really appreciated that. And I also, and just another thing, is just different regulatory groups will accept data sort of at different, I guess I would say, levels, if that makes sense. The U.S. has actually been pretty liberal about results in a dish allowing access to a drug. They've been able to expand the label to really rare mutations based on not studies done in people, just because there's not enough to do this study. Europe's been harder. Is open to that idea, and so we're working with our colleagues in the FDA. But they actually do talk. But I think that's going to be an opportunity for us to leverage why that talk is going to be more important in this new ecosystem. So, thank you. I appreciate that. Good questions. Oh, here we go.
Could you talk to the point, maybe bring us along on the science journey, but when you talk about like Sionna is proving to be better in the lab than even Trikafta was, what does that look like from a lab standpoint? Can you explain to us what that means?
Sure, I'd be happy to start, and if you want to talk about endpoints, sometimes you feel if you want, but you don't have to. Okay. No, I was just going to say, in the laboratory, so one thing we've learned through the experience of all these exciting drugs that have changed the face in CF that Vertex has developed, is that laboratory studies have actually been pretty good at predicting these drugs when they're going to work in the clinic. The screen to develop Kalydeco was actually done in cells expressing a certain CF mutation. Same thing for Lumacaftor, Tezacaftor, all the things that became the combinations today. So we've learned that these studies done in sort of simple systems and then actually in airway cells from people with CF who've undergone transplant, and they've really been pretty good predictors, maybe not on a one-to-one basis, but definitely as a group, for drugs that are going to be active and potentially beneficial in people. And so what we're seeing now is in those models, when you do a head-to-head comparison of something like Trikafta with a combination, as an example, Sionna, Sionna looks better. It gives a higher signal. It might give more, basically, complete normal function. Now, so the first question is, A, does that translate to people? Do you really see that? And B, can we measure that difference? In other words, let's say somebody has a sweat of 30 and another in a drug, and somebody has a sweat of 20. And that sweat's different, but does that translate into less cough, better lung function, better growth? That's the big unknown, sensitivity of the ways to tell that it's actually beneficial. So, yeah, we spend a lot of time thinking about what they call endpoints and how they're changing so much. All the old ones we can't use anymore in the same way. Yeah, was there one? Oh, there we go, back here.
This is a non-scientific question. At next year, yeah, get your glasses on. At next year's VLC, would you consider leading a Zumba class?
Zumba, well, let's, I will. I'm going to warn you, I'm going to wear tights, okay? Just kidding. All right. Any other questions from up there, Patty? All right. Any other questions from the audience that we can answer? I think we're right at the top of the hour, and I think we are. What timing? Thank you, guys, everybody. Really fun. Patty, my darling, thank you.
Thank you for showing up, everyone. And thank you for what you do. Have a great rest of your day.